Hi everyone, my name is Ting Cha, and today I'll be sharing my research project about one bead, one compound combinational library that I've been working on in the Lem lab during the last year. One bead, one compound library design is also known as OBOC. It's a method developed in the Lem lab in 1991 using solid phase split and pool synthesis. The use of hydrophilic surface of resins allowed the beads to be directly screened for compounds that bind selectively to target proteins or other biomolecules. However, recent studies have found that OBOC is prone to give false positive. That means for every bead that we collect as hits, do not necessarily show up as selective or specific binding during the validation process. Every time when a hit is detected, the beads have to be resynthesized again for screening, which can be time-consuming and problematic when the results are biased. Therefore, our main goal is to test out the variety of all resins, linkers, and percentage of peptides on the resin surface in order to optimize OBOC design and minimize non-specific binding. And to do that, we're testing out various types of resin, such as PEGA, PS, and Tendrogel. We're also incorporating stealth peptide sequence, EKs, known as the glutamic acid and aspartic acid respectively. Here in the orange is the target protein, thrombin, that has an active site and 2 exosites. We're using bivalirudin as the ligand for our experiment. Bivalirudin is the specific and reversible inhibitor that binds to thrombin at its active site and exosite 1. Since our target protein is thrombin, the ultimate outcome of the project would be finding the strong thrombin inhibitors. Thrombin inhibitors are used as anticoagulant to prevent arterial and venous thrombosis. Some of these inhibitors are currently in clinical use, while others are in clinical development. The methods that we used were peptide synthesis using Tendrogel because it has shown us better results compared to other resins. The scheme for the 2-layer bead peptide synthesis process is shown here. We use a 2-layer bead wherein the encoding tag resided inside the bead to avoid interference of the screening probe. Then we use gel SDS page electrophoresis to confirm the protein binding by molecular weight, based on the standards given in the ladder. We also use ELISA type assay, where we immobilized our proteins with addition of thrombin and added antibodies to detect the protein. In the staining process, we use a substrate known as TMB, which the HRB enzyme binds to and gives out the blue color effect on the bead. The steps are shown here, where the PBS washes away the excess amount of thrombin and antibody in the assay. The blue color beads imply the binding of thrombin and bivalirudin. In the results section, this is a picture of the gel results. In the very first column showing brown, the latter contains the previously known molecular weight of thrombin, which indicates where it would show up. Because peptides on the resin could also interact with one another due to intermolecular forces that can potentially lead to non-specific binding. So we tested out multiple libraries. The EKs, which are the stealth peptides, are the sweeter ion that does not interact with thrombin and extends the peptides away from the resin surface. Where on the left side, there are 3 peptides in the picture, with the 50% stealth peptides and another 50 with the EK linker along with bivalirudin synthesized. And on the right, there are 3 beads with 50% of the stealth peptides and another 50% with the bivalirudin ligand. And the blue box area indicates molecular weight of thrombin, which is 35 kilodaltons according to the ladder standards. This also suggests that the reading through the bands must have been from the 50% of the bivalirudin interaction with thrombin. There is another scheme where we wanted to test out if down substituting more of the beads to 80 and 20% ratio would give the same results. And we concluded that the 50-50% shows a better binding reading on the gel, suggesting that the 50 and 50 library was useful to reduce non-specific binding. In addition, below in this picture shows the positive hits found after screening the library with 26 nanomotors of thrombin, following by the addition of antibody as described before. As you can see, at position 4, negatively charged amino acids are color-coded in blue. They show similar homology with bilirubin. And at position 10, there are a lot of polarly uncharged residues indicated in green that shows a similar trend in bilirubin. The pink color residues are the hydrophobic aromatic amino acids. We can utilize these sequences to further develop peptides and ligands with higher affinity to thrombin, thus creating better clinical drugs. Thank you for listening. And I also want to take this time to thank Dr. Lam for giving me this opportunity to participate in his research along with Shabnam and Deidre. They have supported me and taught me everything from the beginning. And everyone in the lab who has helped me along the way. Thank you for everyone who came to my poster presentation. Thank you and take care.